Hello guys, I'm here as Meta Alexis with leg number 23 bar 1948-5. So I'm a student of biomedical laboratory sciences in engineering. I'm here on my YouTube channel to talk about the applied immunology. So I'm going to present some more questions related to applied immunology level 3. The first question or the topic of this today is how do messenger RNA vaccine work. The second question is why are adjuvants used in the vaccines. The next question is how are live attenuated vaccines prepared. The next one is what are the advantages of subunit vaccines. So let's start by how do messenger RNA vaccines work. Let's start by asking how do messenger RNA vaccines work. The first is messenger RNA vaccine use messenger RNA to instruct the cells in the body to produce a specific proteins, which typically a harmful piece of pathogen. Example, the spike protein of CARS-CoV-2. The immune system recognizes that protein produced when the messenger RNA instructs the cell into the body. This protein has foreign and produces an immune response, including antibodies. This strain prepares the immune system to fight the actual pathogen if encountered in the future. So, as we know, when the immune system recognize the pathogen or the foreign pathogen, they produce memory cells and plasma cells. So the memory cells remember all the memory cells has ability to recognize that pathogen when that pathogen come in the future. The messenger RNA does not alter DNA and degrade naturally after protein is produced. So here I'm going to show you the chart or the pic that tells you about how does messenger RNA vaccine work. The first, we have the messenger RNA the messenger RNA enters within the cell. For example, human cell. The third step is cell start spike protein production, as I mentioned in the paragraph before. The spike protein was produced. The next is spike protein is recognized by immune system because the spike protein was produced within the cell. The immune system recognized that spike protein produced within the cell as an antigen or as a pathogen. So there is an interaction of lymphocyte. So after the lymphocyte, there is what you call antigen production from B lymphocyte. As we know, B lymphocyte has two cells, plasma cell and memory cells. So plasma cell is the one that is eligible to produce antibodies which interact or which fight against that antigen or that pathogen. So the antibody, as we know, its role is to fight against antigen. So there is immune response. I think this question is well understood. Let's go to the second question, which is called why are adjuvants used in vaccines. So adjuvants are substances added to vaccines to enhance the immune response. As we know, during the production of vaccine, there is a step called the adjuvant. During the adjuvant, we add adjuvant when the antigen we need to enhance the immune response is when we add the antigen, when we add the adjuvant, we add adjuvant to enhance the immune response. So adjuvants are the one which increase the immune response. So they work by stimulating the innate immune system, making vaccines more effective, especially in a population with weaker immune response, as I mentioned before. So example include aluminum salt and squalene based emulsions. Adjuvants also allow for smaller doses of antigen, improving vaccine efficiency and production. So we use this adjuvant when the number of antigens or the quality of antigen to enhance immune response, we don't have that quality or that quantity. So without enough antigen to enhance or to induce immune response, you will add adjuvant to enhance that immune response, which will lead to the production of memory cells, plasma cells, and so on. So figure all the image which will tell you about the paragraph I have mentioned before. Adjuvant will interact with antigen to enhance the immune response. So the next step is when as we have seen in the previous sessions, the cells will when the antigen or the harmful agent enter the cells, there is formation of phagosomes. That phagosome will be processed. So after being processed, there is formation of infrasomal activation, which will lead to the production of cytokines and chemokines. So there is what you call immune cell representation. The immune cell will present or will process that antigens. There is expression of cytokines and the chemokines. So there is a co-stimulator molecules, CD8 and so on, all CD40X. There is naive T cells production. There is the production of T helper cell 1, T helper cell 2. T helper cell 2 will lead to the humoral response. Humoral response is the response which will result to the formation of antibodies. So antibodies will be produced by B cells, which is called the plasma cells, and the T helper cell 1 will induce or will result to the cellular response, which will not include the antibodies. Let's go to the question which is called how are live attenuated vaccines prepared. First of all, the vaccines, as you know, we talked about the vaccines many times, so we are going to see how live attenuated vaccine will be prepared. Live attenuated vaccines are made by weakening a pathogen so that it can still replicate without causing disease. In this paragraph, I'm going to talk about attenuation or attenuated. Attenuated is to weaken or to make pathogen more weak, so that when that pathogen is entering within the cell, within the body, will not cause disease. This attenuation is often achieved by growing the pathogen in non-human cells or under suboptimal conditions. So to make that antigens more attenuated, we will need non-human cells. So we will cultivate or we will grow that pathogen in non-human cells to make more attenuated, which reduce its ability to thrive in human host. Example of live attenuated vaccines include those for measles, mumps, and rubella. These vaccines elicit a strong and long-lasting immune response. So for the vaccine must be strong or must be more stronger to weaken or to fight against the pathogen, or to fight against a harmful agent or infectious. And the vaccine must be long-lasting immune response due to the production of memory cells which comes from the B cells. B cell is the cells which is eligible for production of plasma cell and memory cells. And the memory cells is the one for long-lasting immune response. So this is how live attenuated vaccine is prepared. So in live attenuated vaccines, we will need live pathogen, the pathogen which is living, which is not dead. So during the live attenuated vaccine preparation, we take pathogen which is live, or which is still living, and we weaken that pathogen to not cause the disease within the body. So there is let me make a small difference between live attenuated and inactivated vaccines. In live attenuated vaccines, we weaken the pathogen so that when we inject that pathogen to human being, that pathogen will not cause disease. So in inactivated, we kill totally that pathogen to when we inject that pathogen. So the body will recognize that there is a harmful agent which will come, or which we entered in the body. So here is an image which shows more and clear explanation about how live attenuated vaccine prepared. So first of all is antigenic protein from a wide array of virulent pathogens can be cloned into live attenuated bacterial vector. So we take the antigenic proteins from virulent. So we took the antigenic, the protein which is antigen, which is able to cause disease, and that protein came from virulent pathogen, so can be cloned into live attenuated. So we weaken that antigenic proteins. So the next step is cloning of gene encoding protective antigen, transformation into live attenuated bacterial vector. So there's a formation of plasmid encoded gene, chromosomal integrated gene. Is expression of antigenic protein. So that antigenic protein will be expressed by either constitute promoter or induced by environmental stimuli using any in vivo inducible promoter or oxygen depression. So there is intracellular expression, surface display via signal peptide mediated translocation or membrane or flagella bounded antigen expressed as secretory antigen. So the last is elicitation of humoral immune response. So there is a humoral immune response, there is interaction of, or there is presence of antibodies, and there is elicitation of immune response. So remember that during the vaccine preparation, immunologist is able to choose either immune response or cell-mediated immune response. When you are immunologist, you are the one to choose either I need humoral immune response or cell-mediated immune response. So the next is protective immunity against the target pathogen. So your immunity will be stronger and long-lasting immunity. The last question, or the last topic I'm going to talk about, is advantages of subunit vaccines. Subunit vaccines contain only specific part of pathogen. These subunit vaccines contain only specific part, only one specific part or many specific part. Either you are the one to choose which part you are going to take, such as protein, polysaccharide, peptides, which are sufficient to trigger immune response. You are the one to choose either protein, polysaccharide, peptide of the pathogen, which will trigger or which will enhance an immune response. Advantages: they are safe since they do not use entire pathogen, minimizing the risk of reversion to a variant form. So it means that when you are using subunit vaccines, you take one part, such as protein, polysaccharide, or peptide, not entire pathogen. When you take entire pathogen, that pathogen, when they enter into the body, they can reverse into variant form to cause a harmful problem. They cause fewer side effects compared to live or inactivated whole pathogen vaccines, because these subunit vaccines cause fewer side effects because you took one part, such as protein, polysaccharide, or peptide. They are suitable for immunocompromised individuals. Example include hepatitis B or human papillomavirus or HIV patient, because body will recognize polysaccharide, protein, or peptide more easily than whole pathogen or inactivated whole pathogen to when they enter into the body, because to immune system, when the many pathogens enter into the body, they weaken our immunity, so it will be more difficult to recognize them. So thank you for your attention. Thank you for your your time. So the next video will be more efficient and more good, because during your comment, I need in the comment section you will light what is not going well. So thank you for your time. Thank you for your comment. I will be there for you. Ciao ciao.